Good afternoon, everyone. So before we continue with the lesson on prokaryotic gene regulation, I just want to um, encourage you all to get vaccinated at the earliest. If vaccination is available and accessible as in your location, try to get vaccination at the earliest. Try to encourage your families to get vaccinated at the earliest. Okay. So that you will be protected, and it's not just a service to yourselves but also to the society, so that we'll be able to recover faster from these kinds of problems. Okay, so in the last class we had some troubles, but also we still had some discussion on what is meant by negative regulation, what is meant by positive regulation. That is about the protein or factor. Actually, it is protein that is binding to the DNA, to the promoter operator region. Okay, I'll put it as that. Or I'll say it as DNA. What I actually imply by this is the promoter region. That is the region that is typically upstream of the open reading frame. Okay. So the first in this one, whether if it is if the protein that is binding to the DNA or in that region, promoter region, is a repressor, then we are discussing at the level of negative regulation. The binding of repressor would inhibit the transcription, and the binding or the uh, re- release of the release of the DNA, release of the repressor from the DNA will allow expression. And if positive regulation is whether we are talking about an activator, protein that is, if it binds, uh, it will enhance the transcription or the expression from the gene, otherwise not. But there is also another dimension to this. And the second dimension is in this way, whether um, inducible or repressible, right? And inducible or repressible in this dimension is about a second protein or a factor. A factor can be a protein, it can be a small molecule, a ligand, or so on. Factor that binds to either it could be a repressor or an activator. Okay, but it is about this factor. In the previous dimension, it's about the protein that is binding to the DNA that is present upstream of the open reading frame. So if and what happens because of this binding? It could inactivate or activate, right? A factor, when it binds to a protein, there will be conformational changes, and because of which it could either give a function or deplete or reduce the function of it. So when I say inactive, it is not able to. Not inactive or active state of what? It is the protein that we are actually discussing. It could be either of these. Okay, either it's a repressor or an activator. Not able to bind. The repressor will not be able to bind, or the activator will not be able to bind to the DNA. Right? It is changing the activity. This factor is changing the activity of this protein that we are talking about. The protein could be either repressor or activator. And if it is active, means able to bind. Okay. So in this case, I'll quickly try to summarize what we have already discussed. Repressor becomes inactive when it is bound to the signal molecule. Therefore, it is negative inducible, right? This is the one that we are talking about. Because we say it inducible, because by default the repressor can bind the DNA and repress the transcription, but the presence of signal molecule will inactivate or release the repressor from the DNA. Therefore there will be expression. So by default it is repressed, but now it is inducible. So we're talking about negative inducible. Here we talk about negative repressible, because by default it is active, it is inactive. The repressor is inactive, but binding of the signal molecule will make it active, and therefore it will repress. Right here it is induce. The presence of signal molecule is allowing the induction of expression, whereas here the presence of signal molecule is repressing the expression. I think you can apply the same things to the positive regulation as well. Here positive, because both are positive, because we are talking about an activator, and it could be repressible or inducible. This will be repressible because the signal molecule is causing it inactive, so it cannot bind here. So this is positive repressible. And in the second case, this molecule, the signal molecule, is making the activator active, so it is positive inducible. Okay, try to remember those things. So in today's class, we are going to discuss about 3 or 4 the regulation mechanisms. We'll see how far we get. I hope you have watched the videos, and if you did, we would have certain degree of understanding would be much faster and better. So lac operon consists of 3 genes, 1, 2, and 3. It's LAC-Z. LAC-Z will come many times in your biology, or if you're doing recombinant DNA and other things, and LAC-Y and LAC-A. So because there are 3 open reading frames, there is an operator region, there is also a cap site, that's an activator site, and we call this polycistronic because there are 3 cistrons or genes as an operon, right? All these 3 genes are coded or regulated by one promoter operator region, right? This one. And then first one, the LAC-Z codes for beta galactosidase. This is the enzyme. And lactose permease is the LAC-Y encodes lactose permease, and the LAC-A encodes thiogalactoside transacetylase. So if you see what is their role and so on, at least the 2 roles, two of these is, first one is beta galactosidase. So when there is lactose in the outside, and I hope you know what is lactose, right? Lactose is a disaccharide consisting of gal and gluc. GLC is the symbol for 3-letter code for glucose. So it's a disaccharide of galactose and glucose. And if a cell has to metabolize and derive energy from it, it should be broken down into the monosaccharides, galactose and glucose, and then glucose undergoes glycolysis and releases energy. Galactose is also converted to glucose, and then glycolysis would take place, and that is how you will have the derivation of energy from it. So the enzymes that are coding LAC-Z and LAC-Y, these 2 are, as you say, first thing, lactose is present here outside. The permease is the enzyme that is coded by LAC-Y. The permease will allow the specific uptake of lactose, right? It is kind of transporter of lactose. And then once the lactose is taken up, we need to, it should be broken down into monosaccharides, and that function is carried out by galactosidase. Apart from that, beta galactosidase, you should say that. Beta galactosidase can also do several other functions, like converting it into allolactose. Okay, And there are three reactions, you can say. Beta galactosidase can hydrolyze the lactose to monosaccharides, galactose and glucose, can also isomerize it to allolactose, and can also break down the allolactose into galactose and glucose. So it's like beta galactosidase plays around with lactose like anything, right? Okay. So I hope you understood the components that are involved. The first question is, when do you want the expression of what we should understand also is, when do we want lactose to be produced and when we don't want, right? Lactose, ultimately, you need energy. Every cell requires energy and also matter in the form of carbon and other elements and so on. So typically, if all the matter is already available, then the cell requires energy to exist. Otherwise, it's going to degrade, going to die, right? So what we will think of is lactose is a lactoperon is required for uptake of lactose and metabolizing lactose to generate energy, right? So there you have glycolysis that's already there. That is for glucose will undergo glycolysis and produce energy. You get a net ATP of 2 ATP and so on. Lactose should be converted to glucose, and then it will be metabolized and energy. Lactose and glucose, which one is preferable? If you see, this is an additional step to produce energy. We will come to the discussion. Let's just have a look at the promoter elements and other things before. But keep in mind this thing. Lactose should come into the flow in the form of glucose to the glycolysis, and from there, eventually it should release energy. That is one of the primary objectives of the cell. So the promoter elements are something like this. We have already seen this, I guess. -10, -35, +1, the start of transcriptional, and the region for the repressor or the operator site is overlapping with that of the promoter region. So if the repressor is bound, there is not enough space for DNA polymerase to occupy the promoter. And then also we have a cap binding site. That's an activator binding site. So the promoter, the RNA polymerase, through its sigma factor, will interact with -10 and -35 sequences and will interact with the promoter elements. When does that happen? Only when repressor is not bound to the operator site, that is usually somewhere here. The carboxylic terminal end of the alpha subunit interacts with the cap protein. That is, if the cap protein is interacting with the cap site, and that will enhance the activation or the transcription transcriptional or the expression of lactoperon. So here are the scenarios that I was actually discussing. What is preferable and not? You have lactose, and that should be converted to glucose, and that should be converted to energy, or say ATP. There are also other things. The intermediate products of glycolysis can be used, or also that of lactose can be used to make other compounds or biomolecules. But here we are assuming that the cell has everything and it needs energy for survival. What happens then? So when we have glucose, I will ask the question that I already posed you. If lactose is there and glucose is there, which is the shortest route to generate ATP? That is glucose, right? So It is all about speed. Who is eating faster and growing faster? Bacteria has no other duty, or actually objective of life is to, I mean, different bacteria have different ways, but if you consider E. coli or a species such as those, they have to grow faster. Growing faster is everything for them, because speed gives numbers, right? Means if you grow faster, you will divide faster. Growing faster in bacteria means dividing faster. That is, the speed will allow in the greater numbers. If you're increasing in greater numbers, you're likely to live longer, longevity. And why is this speed also important? You can also think of it in ecological scenarios like this. Say, for example, there is a bacterium or a person is there, and it is a competition. Think of it. And there are several other competitions, right? You can think of each of as a bacteria also. Say A, B, and C. So, and there are 10, 9 units of food. Let's call it as laddu or something, if you like that. And if you divide equally, each of them are supposed to get 3. That is when they are doing it cooperatively and trying to be democratic about distribution. But that's not competition. Democracy is not a competition. Rather, think of it like this. Say this is the ideal situation. Everybody, equality and everything we call an ideal situations. Nowhere in biology there are these kinds of ideal situations ever exist, unless it is the last resort. What would happen if if this person A has eaten 5? What would happen then? The person here, B and C, would get lesser. And say B was competent enough, so the B has eaten 3. Then C is going to get only 1. So eating fast is twice good. I'm talking in bacteria, okay? Consuming more or consuming quickly is twice good, because say, for example, here you're growing better, you're empowering your own existence and so on. By doing so, you are also, by eating faster, you are depleting the resources that are available to B and C, right? You are getting rid of competition, okay? And that is why in most of the bacteria, if not all, growing fast is very important. And that's why whichever is able to consume more in the shortest span of time will divide more and will outnumber the others. And that is how competition always plays in ecology. It's like what humans do. We assume A is humans, right? We are consuming everything. Almost we ventured into land, sea, and air, and also we are now looking at Mars. We are trying to use up all the resources. What is happening to the other species then? There is lack of space, lack of food, lack of many things for their survival. And that is how a large number of species are going extinct in this on the surface of Earth. And our activities actually cause that. I hope you understood why consuming more is important for bacteria, at least in true ecological sense. That's actually the law, that be fast, consume more, and reproduce more, so you'll outnumber the others and get rid of the others. So if there is lactose and glucose, what would you like to eat first? Do you want to turn both, or which one is the easiest food? It's glucose. In that scenario, would you want lac-operon to be on or not? I'm only giving scenarios. I think these two are easier to answer. In the first stage, I'm saying glucose is available, but lactose is not. do you want lac-operon to be expressed? What are the genes that are for what are the enzymes that are produced? Beta galactosidase, permease, and so on. They are they will be produced, say for example, and what will they do? And in the last lesson we have learned the expenditure of to produce a protein from mRNA. Does it make any financial or economical sense to produce the mRNA of lac-operon, that is 3 genes, and to produce these 3 proteins or enzymes, and actually each of them would be multiple copies, right? Because transcription, if the promoter is on, then you'll keep on, RNA polymerase will keep on producing the RNA. And if the mRNA is present, translation will keep on translating it. And what will these proteins do if lactose is not present in the cytoplasm? It's absolute waste. There's a lot of expenditure for these to produce all these things. A lot of ATP and energy is produced. And you also, because lactose is not present, these 3 proteins are useless. You have to degrade them. That is another expenditure, right? In these scenarios, this is, of course, this is the essence of regulation. It is to reduce unnecessary production of proteins in the cell, which in the resources could rather be used for some other proteins or production of some other essential stress proteins and so on. So in these scenarios where lactose is not present, the best thing is to have no transcription at all, so that you don't have to waste your expenditure on these things. And that is when repression actually happens. Think of the other way. There is no glucose, but lactose is present. What do you want it? There is the shortest route to the energy, as I was telling you, is lactose, and then glucose, and then ATP, or say energy. So if glucose is not produced, is not available to consume, then what is the next best solution? Then it is lactose. From lactose you can generate glucose, and from that you can generate energy. So in that situation, you will need to take up the lactose that is present outside, and then get inside, that is broken down into galactose and glucose, and then glycolysis to ATP. So in that situations, you want high level of expression from lac-operon. This is what I was telling you in the first scenario, where both glucose and lactose are present. Say both are present. You still want glucose because it's the easier route. The ones, if say in bacterial experiments that we do in the lab, usually you take a conical flask or tube, you provide the medium nutrient broth, along with you inoculate, you add a few bacteria, and you can put in glucose or lactose as you want, or both. And in those cases, what will happen? That is how studies are usually done. In this scenario, you don't want lactose operon to be actively transcribed, but it is available. So it's kind of being ready for, once the glucose is consumed, you consume glucose first, and then be ready to consume lactose, right? And in this case, you will have very minimal level of expression, very little bit. We'll see the scenarios in the next slides, okay? But before, I hope you will also try to remember something called as XGAL and IPTG, isopropyl thiogalactoside. These 2 compounds are used in labs for practice in experiments with bacterial genetics and so on, even in recombinant DNA technology for expression of lac promoter. So this is lactose, and this is the substrate for beta galactosidase. Beta galactosidase can also work on something called as XGAL, a compound, and it will break it up, and then this compound will give a blue color. Okay, when we do the labs, we will have plasmid isolation and transformation experiments. In those, these might get useful. And there is something also, allolactose. I hope you remember. Galactosidase can, beta galactosidase can convert lactose to allolactose. Allolactose and lactose, they differ in the carbon, say 1, 2, 3, 4, 5, and 6. This is 1,4 linkage, right? In allolactose, you have a different one. It's 1, 2, 3, 4, 5, and 6. You have 1,6 linkage. Okay, so it is just an isomer of lactose where the bond is, the glycosidic bond is little different. The from the fourth carbon, it is now on the sixth carbon. Allolactose can act as an activator, which we will discuss in the later slides. First, the lac repressor. There are three regions for three operator regions like this. Typically, this is the most important one, where lac repressor binds, and then it can form a tetramer like this, so that this loop will prevent any activation or transcription from lacoperon. Okay. Yes. Let's see this, and then we'll go there. First thing is what happens. I hope you can see it clearly now. What happens when lactose is not present in the medium? Okay. And everything is fine. Here is lac P represents lac promoter, and lac Z, lac Y, lac A, and here you have lac I. Lac I is the gene that produces a protein, that is the lac repressor. Lac repressor is not a part of the lacoperon. It is being produced somewhere else in the genome. So the lac repressor will bind to the operator region that is overlapping with the lac promoter. And if lac repressor is bound, then RNA polymerase cannot interact with the promoter. So there will not be any expression from the lacoperon, no transcription. Okay. This is when, in the absence of lactose, and this is how repression is achieved. There will be production of lacoperon, lac repressor, and repressor is active by itself, and it can inhibit the transcription from lacoperon. Let's see what happens in the presence of lactose. If you find that there are too many things, that's all right. We will try to have an overview all the time. But you can also watch several videos. Lacoperon, tripoperon are quite popular and quite common that most people study all the time. You can also visit a lot of YouTube videos for better understanding, or animations probably. They will be of help. So when there is lactose, something is going to change. So transcription is producing the repressor, lac repressor, but the repressor is bound by a compound like ligand, and that is allolactose. If allolactose binds to the repressor, repressor becomes inactive. That means it can no longer bind to the operator region, which would also mean that RNA polymerase can now interact with the promoter and undergo some activation, right? Or the transcription or gene expression from lacoperon would happen. So how is allolactose produced? It is the first question. Where is the, yeah. Let's go in the order of what is described here, so that we will have a better understanding. So when in the presence of lactose, there is a production of allolactose. So how did allolactose come? The allolactose is from the lactose that has been taken up. Why is allolactose? If you're saying previously there is no lactose, now we have lactose, and you're already saying there is production of allolactose, and that binds to the repressor, and repressor becomes inactive. Where did we get allolactose from? It is possible if only there is lactose in the cytoplasm. But then we added the lactose outside. How did it get inside? To get inside, you need lactose permease, which is a part of the operon, lac operon. When were these proteins produced for the uptake of lactose? That's the question. So, as we said, there is always basal level expression of lactose, lac operon. There will be little bit minute quantities. So, you'll always you can expect that some permeases are produced. And if they are there, they will take up lactose a little bit, very low in level. And that lactose will be converted to allolactose. And if here we are, so allolactose is produced. And again, for the conversion of lactose to allolactose, we need beta-galactosidase. Where did that come from? Again, that would be from the basal level production of it. Okay. So the allolactose will interact with the lac repressor and inactivate it, so it can no longer bind to the operator region, which will make space for RNA polymerase to allow active transcription from the lac operon. That is when you'll have the production more or higher level production of beta-galactosidase, permease, and transacetylase. And they will continue to take up lactose and degrade the lactose to glucose and galactose, and then they will enter the glycolytic pathway. Okay. So I put this figure so that you will be able to appreciate what are we talking about. Is it positive regulation or negative regulation? We are mostly talking about repressor. So we are talking about negative regulation. And so the protein that is interacting with the DNA here is repressor. And that's why it is negative regulation. Is it inducible or is it repressible? That's the question. How do you determine whether it is inducible or repressible? That is about what does the ligand do. The ligand here is allolactose. And allolactose is, if the allolactose is not present, what would happen? By default, it is going to get, repressor is active in state, and it will interact with the DNA operator region and bind and inhibit transcription. So the presence of allolactose is allowing the lac operon to be expressed, right? Induction of expression. So it is negative inducible operon, lac operon. Okay. We'll also look into the other one. There is also positive regulation about it because we spoke about cap site. We will talk about it now. And then maybe in the next class, again, we will try to revisit it so that you will find it comfortable. So there is, lac operon can also be under the control of positive regulation. When you say about positive regulation, we are talking about an activator. Right, and the activator here is called as CAP, catabolite activator protein. And CAP binds to the CAP binding site that is present somewhere upstream of the lac promoter. So when glucose is high, when glucose levels are high, that means we spoke about this already, where if glucose is present, that is, glucose is the preferred metabolite, the preferred energy source. So if glucose is there, we do not want so much of lac operon to be expressed because we want to use glucose more because it's economical. When there is high concentration of when glucose is low, we'll see a scenario what happens. In glucose is low, you will have high levels of cyclic AMP. It's a kind of signal molecule. Cyclic AMP is derived from ATP by adenylyl cyclase. I hope you remember we use adenylylation as a word when we are talking about charging of or activating amino acids for charging of tRNA. Adenylation meant that an AMP is added to an amino acid, right? AMP, adenosine monophosphate. Here, adenylyl cyclase, the enzyme, is acting on AMP or ATP, or it is making AMP, which is cyclic. Why do we say it's cyclic? Typically, the phosphate is attached to the fifth carbon here. Sorry, 1, 2, 3, 4, and this is the fifth carbon. The phosphate is attached to that and not to this one, right? Here you would have 3' OH, isn't it? The next phosphate would be bound here if it is in DNA or RNA. So basically, when you have a bond formed between the phosphate, the alpha phosphate, and the 3' OH, then you call it is a cycle like this, right? It's a kind of continuous one, so you call it as cyclic AMP. I hope you understood that. And that is done by the enzyme called as adenylyl cyclase. Okay, so this cyclic AMP is can interact with CAP, and CAP, now if it is bound with CAMP, becomes active and it binds to the cap site here. And if it is binding to the cap site, it will enhance the production or the binding of RNA polymerase to the promoter site, and it will increase the production from the elacoperon. Okay, so let's see the other scenarios also. When glucose is high, okay, when glucose is high, what do you want? You don't want expression from elacoperon typically. So the CAMP levels are usually low. That is another pathway about how high glucose concentration can result in increase or decrease CAMP. But we are not bothered about it at this time, because what we should remember is high glucose level, low CAMP, low glucose level, high CAMP. They are inversely proportional in a way. So when glucose is high, cyclic AMP levels are very low, because of which they do not interact with cap protein. Because they do not because the ligand is not present, cap is inactive and cannot interact with the cap binding site. Therefore, there is no activator, so the binding of RNA polymerase is not very efficient, because of which there is very little or some expression. That is a very low rate, and that is called the basal level expression. Is there anything bound to this DNA? There is no there is no activator binding, there is no repressor binding, and even in that cases there is very little amount of transcription that happens, and that is called as basal level. Okay, so we will try to again do what we have done. Is it negative regulation or positive regulation? It is positive regulation, because we are talking about an activator, and that is called as CAP. And is it inducible or is it repressible? That's the question. And then inducible or repressible, we are talking about the signal molecule. The signal molecule is called CAMP, cyclic AMP. So what is the presence of cyclic AMP? What is it doing to CAP? To be active, CAP is active only when it is bound with CAMP, right? So if cyclic AMP is not present, CAP is inactive, and then it cannot bind. That is the default state. If CAMP is not there, CAP cannot interact with the cap binding site. So what is happening in that case? Do you expect expression or no, or repression? There is, or you can say it as no expression. So it is actually kind of repressed. But the presence of cyclic AMP will activate CAP and then will allow drastic increase in transcriptional activation. So it is positive and it is inducible. The previous one that we have seen is negative inducible. Okay. I hope you understood what is. Try to keep this in mind because it's a recurring phenomenon in all prokaryotic transcriptional gene regulation. I'll give an introduction to tripoperon and we'll see how far we get. Here, the objective of tripoperon. What is the objective of lacoperon? Let me try to. Lacoperon is to hydrolyze or metabolize lactose. That means the lactose that is taken up would be degraded. Tripoperon is reverse. Tripoperon synthesizes or produces tryptophan. Okay, it is the objective is to, it is a biosynthetic operon. Tryptophan is an important amino acid. It's a charged amino acid or polar amino acid, and then you would need to have the cell needs to produce amino acids. So say for example you're only giving lactose or glucose, but to build something you need amino acids. The cell, unless you're supplying from the outside, the bacterial cell has to prepare or make all the amino acids by itself, utilizing the energy matter that comes from the outside, right? So they have operons to synthesize proteins or enzymes which synthesize tryptophan. Typically, each of them is produces one enzyme, and that enzyme is responsible for in the pathway to synthesize tryptophan. If you read that in biochemistry, you might try to associate this knowledge with that. There is a repressor and a co-repressor, and there are two levels of regulation in tripoperon. One is at the transcriptional level, and the other one is called attenuation. And that is where this is why attenuation is an important or smart concept that we will try to know, and that is a complex also. So if you understand that, it will be a nice way. That is why tripoperon is important. So I'll try to give an introduction to these. So I'll just skim through so that you'll get to know some facts, and tomorrow we'll have a look at them. Okay, so there is a let me talk about the components. Here you have the operator region. This is the operator region, and you have the promoter region here, and this is called as the 5' UTR or untranslated region. And you have several structural genes, TRPE, the untranslated region is, okay. Untranslated region is probably small here. And then you have TRPE, TRPT, TRPC, TRPB, and TRPA. All these are enzymes that are important to convert the crocimate to tryptophan. So there is a repressor, and this is the gene that produces the TRPR. TRPR for repressor. It produces an inactive repressor. This inactive repressor cannot interact with the operator region, right? But if the tryptophan, when tryptophan is high in concentration, so sorry, here we'll talk about this first. When tryptophan is low, here the ligand is tryptophan itself. Okay, the TRP repressor is inactive if tryptophan is not high in concentration. Okay, it is low in concentration, so it is inactive repressor. So it cannot bind with the promoter. So what will happen is transcription will happen, and the production of enzymes from which will convert correspond to tryptophan. But if you have if tryptophan is high within the cells, this is in this cell, okay, not like lactose outside and so on. Anything detection can only happen of what is present in this cytoplasm. So if the tryptophan is high in concentration, it will bind to the trip repressor, and because of which now the trip repressor will become active and it will go and bind to the operator site here. If it binds to the operator site, the transcription, the RNA polymerase cannot bind here, and therefore you will have you will not have transcription from this one. It is very simple, very similar to that of what we have discussed with lacoperon. The next level is a little bit complex, but before going into that, I want to also discuss, is it positive regulation or negative regulation? It is negative regulation because we are talking about repressor here. And second thing, is it inducible or repressible? By default, it would be repressed. By default, it is expressed. And the presence of the ligand is causing the repression. So you can call it as negative repressible. This is negative repressible regulation. Okay, the next thing we are trying to learn is attenuation. This is just introduction, so we'll feel comfortable in the next classes. So the trip operator region, there is a leader RNA, right? And the leader sequence or leader peptide, I'll try to explain. I'll see if there is one here. Yeah. So assume that the lacoperon is something like this. Sorry, trypoperon. It has several of these genes loaded. And here is the transcriptional site. This 5' untranslated region is also called as leader region. It has an important regulatory function. Within this leader region, there is a small leader. You call it as strip L. That will, if it is transcribed, will produce a leader peptide. Okay, it does not have any big functions. It's something similar to that of upstream open reading frames that we learned in translation of eukaryotes, variations in translation of eukaryotic genes, some of them. So at the level of RNA, you will think of it as RNA like this and assume that I will try to, okay, assume that this part of it is the leader. The other ones are here you have the open reading frames of other genes. This leader region is what is depicted here, right? So from here, AUG till UGA, that's the stop codon, right? The mRNA is produced. This is the 5' end of it. And so I'm indicating this part of it. And here is the first structural gene that would be translated, or the open reading frame starts. Within this leader region, or the 5' untranslated region, you have certain regions. You call it as region 1, region 2, region 3, and region 4. Why are we even calling them? Is they have certain degree of complementarity. So that means they can form hairpin loops like this. 1 and 2 can form hairpin loops. 2 and 3 can also form hairpin loops. And 3 and 4 can form hairpin loops. There are several possibilities. So if you have, let me put it here. 1 and 2 can form. 2 and 3 can form. 3 and 4 can form. If 2 and 3 have formed a hairpin loop, 1 is individually separated like that, and 4 is separate. They cannot, right? Because 4 can only base pair with 3, and 1 can only base pair with 2. So if 2 and 3 have base paired, 1 and 4 will be like that, right? But think of it as if 1 and 2 have base pair, what will happen then? This is not going to happen, but this will happen. So 1 and 2, if they base pair, then 3 and 4 will automatically form. Right? So that is how you can think of. During when the situation, there are also certain things that I need to describe before I go, and I will stop there. There are two, you can see this one. There are two GGU, and I think, is it right? I think it's UGG, UGG. Those are two tryptophan codons that are present within this leader peptide. And that tryptophan codons are present in region 1 here. Okay. So if a ribosome is translating from here, I'm starting here, or I'll describe this one and I'll stop, so that we'll discuss the rest in the next slides. Assume a ribosome is loaded here and it is translating here. And when it comes to this part, right? And if you're saying tryptophan is low in concentration, what will happen? It will get stalled, because there are no charged amino acids with tryptophan, because we are saying tryptophan is low in concentration. Then what will happen? Which one will form? 1 and 2, 2 and 3, 3 and 4. This region 1 is occupied by the ribosome. So it cannot base pair. It will not allow the 2 region to base pair with 3 region. Right? So the hairpin loop cannot form. It is something like a situation like in this case. Ribosome is translating and it is stuck or stalled within the region 1. So what will happen? 2 and 3 will base pair like this. And then the mRNA, just we'll look at what is forming or not. In the next class, we'll see. If you have enough of tryptophan, what is going to happen? Ribosome, in this case, because there is charging, it will just keep on moving. It will occupy 2 and 3. It will just move very smoothly towards this region. And then you will have 3 and 4 forming a hair loop. In this case, translation is inhibited. If there is no protein synthesis, that means no ribosomes are actually at all loaded at all, then you will have 1 and 2 and 3 and 4 hairpin loops formed. It's okay if you feel confused about it, but these are some things that you need to remember, try to understand. In the next class, we'll try to look at it. Okay? So in the next class, I think I'll move a little faster. Please watch the videos before you come. We'll talk about these 3, and then we'll go into phages. Okay? Take care and have fun. Please get vaccinated at the earliest, so that you're all safe.